So these questions are more for review. We're going to go over them very easy because if you took introductory biology, I'm sure that you know the answer. So first question, let's read it together. The cell cycle consists of the following phases. So G1, that is the first control. So they go to synthesis. So here, the most likely that is synthesis. G2 is to know that the DNA replication went well. And M is for mitosis. So here it should be synthesis, meiosis. Um, here is incorrect. Prophase, remember, I'm going to repeat it multiple times. Prophase, we're going to discuss different topics in this course, and one of them will be we'll focus on. I know you have heard multiple times about mitosis before. In this course, we're going to talk about prophase, that is the stage where the crossover will take place. Yeah, and it will be divided in 5 sub-stages. Metaphases is one of the stages of mitosis, so this is incorrect, and G0 is another stage of control. So the best answer here, I'm sure that is that you know that is synthesis. That is the synthesis, the replication of the DNA. So here, as we said before, so all of these steps are called interphase, and in the interphase you have these control phases, G0, G1, G2, and in between that you have this S, that is the synthesis where the chromosome is duplicated, and here you have you have here the mitosis, and in this course I'm going to tell you that here you can subdivide it in 4 more stages where the crossover takes place. So that, as I said before, this is wrong for this question, and this is wrong. So let's go to the next question. Again, a pretty easy question. Let's read it together. The process of cell division that ensure that each of the 2 daughter cells. Okay, so here we have 2 daughter cells. So this is a key piece of information. If it was meiosis, you will have 4 daughter cells. And these 2 daughter cells receive a chromosome complement that is identical to the complement of the parent cell that is known as. So second piece of information that is important here is that receive a chromosome complement identical to the parents. So here again, they're showing you that this is not meiosis, because in meiosis you will receive half of the cell, half of the number of chromosomes. So here this will be wrong. It's not meiosis. Replication is a different thing. Here this is they are asking you for the process of mitosis, not for the process, the name of the process, how you call the process of the duplication of the DNA. So best answer, I'm sure that you know, is this one. In this course, we will talk about this disjunction and non-disjunction. That is when the chromosome separates, and we will cover them more in chapter 5. Okay. And today, again, the magic word for this week will be crossover. Remember, take place in prophase 1, and there are 5 subphases, and we will talk about that. But in this case, the answer will be mitosis. So here is not crossover. So again, mitosis is correct, and I already told you why the other ones are incorrect. Okay, this is another easy question. Let's read it very quick. The portion of the cell cycle during which the chromosomes are replicated is called. So this is for control, as we said before. This is a phase for control. This is the M is for the mitosis. Prophase is where, again, so where prophase 1 will be where crossing over take place, and is divided in 5 subphases. And the best answer here, it will be S. That is from synthesis. So here, S from synthesis, and we already talked about this, and I'm not going to talk anything else because you already know the answer. In the following question, I'm going to cover a little bit more of material because it will be new information. So now we're going to read this question that is just a true or false question. Crossing over at the chiasmata occur during prophase 1. Okay, so I have told you during the previous question that crossover take place during prophase 1, so I already gave you the answer multiple times, and we're going to talk a little bit more about that. So we know that it's true, and we're going to define terms of what is crossing over, and what is chiasmata, and what are the subphases of prophase 1. So let's do it together. So you already know that this is that crossing over take place in the prophase 1, and now I'm going to quickly review some aspects of the cell division and answer from mitosis and meiosis, focusing on crossing over. So at this point in this course, I'm sure that you're expert on mitosis and meiosis. What I'm going to tell you is that in this step, something very interesting happened. Yeah. So before the chromosome have separated and go into different cells, here the crossing over take place in prophase 1. Yeah. Here, although here we are focusing in that they should look like this. Actually, they should have pieces of the other. Yeah, this should have here a red portion and a red portion, and here you should have a blue portion, yeah, of the chromosome. And this start happening since here, yeah, that you start mixing, doing crossover. But in this diagram, it's not shown because this is not the main point of some of these ideas of mitosis and meiosis that they were explained in previous course, and now we're going to talk about that. So here, as I said here, this is the meiosis, and in meiosis 1, you see that there is a separation of the homologous chromosomes. Yeah? So here in this stage, but I want that you see that the chromosome here in your textbook, I think this is taken from actually a kind of biology. You see that the chromosomes are not completely blue or not completely red in this telophase and cytokinases. Yeah, they are actually a mix. Why? Because recombination already happened. Here in anaphase 1, it was already happened, and here is when the process it was almost done. You cannot see clear the difference because of the drawing, but all this start here. Yeah, this crossing over start here, and here you find key information that maybe is new for you, that this process of pairing of the homologous chromosome, very important, that and this pairing is the alignment of the chromosomes, yeah, and that form this physical structure that is called chiasmata or chiasma. That is, you have a lot of proteins that they form like a bridge and allow these two chromosomes to, in a safe way, exchange pieces of DNA. Okay, so let me tell you more about this stage of prophase 1. So now we're going to talk about prophase 1. And here in this diagram, you can see the different stages of prophase 1. So in the beginning, I have to tell you that prophase 1 is the longest phase of meiosis, and there are three main events. So the first event is the condensation of DNA. So the DNA, it was already replicated before in the phase of synthesis, and then now you need to form the chromosomes. After the condensation, the second stage is the synapsis or physical contact between homologous chromosomes. So here you have specific proteins here that are going to not only help to align, yeah, to create a perfect alignment, but to create a bridge that it will allow to cut and exchange information. And this is the third stage, that is the crossing over, where the genetic material is exchanged. So remember, synapses occur before, that is the alignment, and the crossing over is the exchange of the material. And the last stage, when the chromosomes have different portions, yeah, as you can see here, that here you have this blue, and here you have this red. That they start mixing the chromosomes. That will be called recombination, the final process. Okay, so I will explain that in these three terms: synapses, crossover, and recombination, that are similar. They seem very similar, but they mean different things. And in the following slides, I will cover that in more detail. Now, uh, just to finish this section of prophase 1, I want to show you the 5 subphases of prophase 1. So maybe you haven't heard of these 5 subphases that are focused on the crossover. So I'm going to talk about this, and you can review that in your textbook for more information. I'm going to try to pronounce it, but I'm not going to do a good job. But so here, the first stage is leptotene, here's zygotene, pachytene, diplotene, and finally the fifth is diakinesis. Okay, so what happened in the first one, in leptotene, what is happening is here the prophase begins, so the chromosomes start to condense. So here in this stage, there is no crossover yet. In the second stage, the zygotene, what is going to happen is that the synapses, the alignment, is going to form, and so you need these complex proteins, yeah, that are going to help this attachment, this process of alignment in the synapses. The third stage, this pachytene, that is where actually the crossover takes place, and there is an exchange of the non-sister chromatids. Finally, the diplotene is when the synapses ends, and here is when the chiasma are visible within the bivalent chromosomes, that this combination of two chromosomes. So just to remind you, here is the key step. Let me put it. The pachytene, here is where the magic happened, and here is when the chiasma, you can see it. Yeah, the physical exchange. Yeah, where you see that when one chromosome seems to be on top of the other one. Yeah, so that is important. But here the crossing out already took place. That this could be a question for the test, and for a question in the quiz too. So diakinesis, here is where the prophase ends, and here in this stage the nuclear membrane already disintegrate, and here if you see, this is already, the exchange already took place because it took place in pachytene. Okay, so remember 5 different stages. The first one is when the prophase, the chromosomes start condensing. The diakinesis is when um, the exchange already happened and the nuclear membrane already disintegrate. Here, the most important, and that it could be in the test, is pachytene, is when the crossing over actually take place. Okay, so now let's review the difference between synapses and crossover. So here you see many concepts that you have here before, the kinetochore. So here allow the centromere, allow the central polar part, or it could be more the region where the chromosome is being pulled, and the kinetochore is where all these proteins that will allow the pulling, whether they will be located. And you have here, so you generally hear about this one. Then you have here about this homologous chromosome, the blue one here and the red one, that are the homologous chromosomes. Here now are the new words. The this synaptonemal complex. That is the one, if you see these white structures represent proteins that are going to help to align both chromosomes. Yeah, that is very important for the process of synapses. So synapses refers to the pairing of the homologous chromosome during prophase 1. Okay, so the synapses is pairing. Well, crossing over refer already to the exchange of the genetic material during synapses. So synapses occur first, and then they will be the exchange of material. So for that reason, they said that synapsis is followed by the crossover, or while crossing over is preceded by synapsis. If there is no synapsis, if there is no pairing, if that complex of protein that attach these two chromosomes align it properly, there will be no proteins that allow to create these bridges to cut and exchange material, so crossing over is not going to take place. And in the following chapters, we are going to talk about what happens when you have chromosomal rearrangement to be able to have crossover, or what could be the problems when you don't have this proper crossover. And the synapses, this pairing always occur, yeah, but it doesn't mean that crossing over will always occur. Yeah, many times crossing over doesn't occur. Yeah, it occurs only in specific places, while synapses always occur. Okay, so that was an important topic. Difference between synapses and crossing over. Hopefully, it was more clear now. And now we're going to talk about another topic uh, that maybe you remember from the class. That is the chiasma and chiasmata. So here you have a chromosome. Here you have the diagram. Yeah, and in this you have not only one chromosome, you have two homologous, one here and the other one here. And it's much clearer when you go and see it in the diagram. One chromosome here, the second one chromosome here. So you remember the centromeres, those sections of heterochromatin, yeah, that are just repetition of DNA that help you to pull the chromosomes. So random containing genes, and they contain repetitions of DNA. So here you can see the centromeres here. And in addition to that, I'm going to add on purple these structures that you see here. Yeah, that for a non-expert eye, they said, "Oh my God, you have here uh, like all type of centromere. So what is happening here?" When you in the diagram, they show you much clearer what is happening. So here you have the blue chromosome, and now has a piece of the red chromosome, and I will come back to the blue one. Yeah. So what happened here is that between homologous chromosomes, yeah, there was a structure, yeah, that formed between these homologous chromosomes during by the recombination, and this is a physical link that occurred during mitosis. Here you see this word chiasmata. This chiasmata means the plural because you have one here and two here, and in the diagram you see one here and two here, so you call this chiasmata for singular chiasma. Okay, so very important, it's a physical structure that form as a result of the process of crossover. So now, just to avoid confusion, we're going to define again synapses, crossover, and recombination, and with that we finish this question. Okay, so remember, synapses is the pairing of the chromosomes. Yeah, so this is pretty easy. Now I'm sure that you know that. Uh, Crossover refer to the exchange of the materials. So synapses, during the synapses, there is just pairing occurs all the time. Crossover occurs less times and goes after the synapses, and is the exchange of material. And so here you see the the alignment. Yeah, here the crossover, and finally here as a result of when the crossover already finished, you find the recombination, that is the production of different combination of alleles in the offspring. So the result is called recombination, and that is different from synapsis and crossover. Yeah, so these three topics sound similar, and but are not identical, are not synonym, but are related to each other. Okay, and with that, we finish all the questions of the quiz. So that super extensive extra explanation, just to remind you what it was. Now you should know what is crossover. You know that this is different from chiasmata. Yeah, you know the difference between crossing over, synapsis, and recombination. You know that chiasmata is the plural. You know that the singular is chiasma, and you know what is this structure, and you know what is prophase one, and all these subphases. So you know that crossover, the chiasmata occur during prophase one, and this is true. Okay, I think with this information, we are done for the quiz. If you have any questions, just let me know.